Since at least the 18th century, we have been fascinated with cutting and pulling small animals to pieces. So in the 1700s, Abraham Tremblay bisected a Cnidarian polyps again and again and again, and generated a 7-headed monster, which he aptly called Hydra. In 1900, another one of my scientific heroes, Thomas Morgan, worked with the small flatworm planarians, and he determined that as small as 1/279th of the piece of the animal can still regrow into a full worm. And finally, we have these beautiful experiments in newts, axolotls, and salamanders that demonstrate that even some vertebrates can regrow limbs. Unfortunately, not all of us are that fortunate. So mammals and birds in particular are the poorest of the poor regenerators. Right? So humans, like Kristin studies, can regrow quite a large portion of their liver. And then children, reportedly, but please don't try this at home, can regrow fingertips and the tips of their toes. Right? But not us anymore. That we lost that ability as we grow. But of course, this ability pales in comparison to what planarians, hydras, or salamanders can do. So why do some animals regenerate well and some others don't? There are at least two views historically in the field for this question. Some biologists, including Charles Darwin and August Weismann, hold that regeneration is an evolved trait. It's a trait that evolved for a specific organ for adaptive purposes. So for instance, lobsters may have evolved the ability to regrow their claws because they lose them often in mating or foraging. Some other biologists, including Thomas Morgan, held that regeneration may be inherent in all of us, just as cell cycle is inherent in cells. In the lab, we're intrigued by Morgan's view, because if regeneration is inherent in animals, then could we go to animals that normally poorly regenerate and coax them to regenerate? So to play with this idea, before that, as you can imagine, this is a long debate, and resolving this debate remains a challenge to this day. But thanks to more and more interest and also just the ability to study non-model animals this past decade, we have come to a realization. So here is the animal tree. Here's us in the phylum Chordata. And what we've realized from studies of many in the field is that many animal phyla have one species or more that can regrow quite significantly their body parts, which means that regeneration is widely distributed in the animal kingdom. Regeneration being widely distributed have led to some in the field to argue that perhaps regeneration is ancestral in animals. Of course, this is still a discussion in the field because we still cannot rule out convergence. We all know convergence can be quite common. But it does raise the idea that perhaps if it's ancestral, then can we promote regeneration? To approach this question, in the lab, we work with animals that, like humans, poorly regenerate and ask, how can we promote regeneration in these animals? For now, in this talk, we focus on appendages. Few in the fields have pursued how to stimulate limb regeneration, and the results are quite encouraging. So in the frogs, studies that peaked mostly in the 20th century, and then there's some recent ones in the past 5 years or so, show that limb regrowth to different extents can be stimulated in adult frogs using various means as simple as just applying salt or electricity or implanting progenitor cells. This one was from Jonathan Zleck's lab In mouse digit, so mice, just like human children, can regrow their digit tips, but nowhere below the first phalange. It has been shown that digit regeneration can be coaxed by applying developmental signals into the cut digits. So as you can see here, that even though it's encouraging, one thing that we notice is that different species, you have different methods that work, different molecules that could coax regeneration. Since we're intrigued by Morgan's idea that regeneration might be ancestral or inherent in animals, in the lab we wonder, can there be a conserved mechanism to promote regeneration? And the reason we're interested in this question is because we think if we can find something that works across species, then that can give us a little bit headway into understanding the evolution of regeneration. What factors might have influenced the evolution of regeneration in some species and not others? To study the question of whether there is a conserved strategy to promote regeneration, we reason that if there is a conserved mechanism, it is most likely more impact in the basal lineages. And for that, we study the cnidarian, the moon jelly. So this is one of my favorite pictures, the moon jellyfish is taken in the waters of Southeast Asia. In the wild, so this picture is an optical illusion. The moon jelly grows to be about a foot in diameter. And we studied the juvenile stage called the ephyra. So this is a real-time movie of an ephyra swimming. It is about 3 millimeter in diameter. And we study, you see these 8 appendages organized symmetrically around the body. They're called arms for simplicity. And they are swimming appendages. They pulse synchronously and generate fluid flow that facilitates feeding and propulsion. So the first thing that we did when we had the jelly in the lab is what my student wanted to do with every little creature is to cut them to pieces. But then he did that, and then he came running to my office and told me, well, actually, to my surprise, the jellyfish did not regenerate. So what I've shown you is experiments mostly in ephyra, but we have seen the similar results in the medusa stage as well. So here is an ephyra cut into 2 pieces, a 5-arm piece and a 3-arm piece. Here's what my student saw. In 12 hours, in 1.5 days, and in 2 days. And he observed the same phenomenon with the other piece. So instead of regrowing lost appendages, reorganize existing appendages and regain radial symmetry as quickly as within 1 and 2 days. The recovery of symmetry was robust. We could perform various amputation schemes and generated a 2-armed ephyra, 3-armed ephyra, 4, 5, 6, and 7-armed jellyfish. More than 90% of the ephyra recovered symmetry. And those 90% of the animals continue growing and mature into medusa. The ones that failed to recover symmetry grew abnormally, and these guys don't swim. They just stay sunk at the bottom of the aquarium. So this is what we reported in 2015. The moon jelly ephyra respond to dramatic injuries by reorganizing existing body parts and regain radial symmetry. We observed this in multiple species of jellyfish. And then we showed that this process does not depend on cell proliferation, does not require cell death, but it is driven mechanically by forces from the propulsion machinery. So that was in 2015. But curiously, After having cut thousands and thousands of ephyra, we start observing. Sometimes we would see a little bump at the cut site. And then more rarely, we would see something elongated from the cut site. So that made us wonder, what's going on? So we repeated the experiments in the natural habitat where our polyp population was collected, which is just off the coast of Long Beach. We repeated the same experiments. We removed 3 appendages, and we drove to Long Beach, which is an hour away from where we are. And here's a picture of our custom tank, and then just Nalgene jars, but we replaced the top with we put filters on so water can flow in and out. We put our jellyfish in the Nalgene jars, put the jars in the tank, and lower the tank into the water. 2 weeks later, we came back and pick up the tank and look at the animals, and this is what we saw. Most of the animals still regain symmetry, like we saw before. But then in 2 out of 18 animals, here's what we saw. There's a small arm growing. Right. So this is 2 weeks after amputation, and then this is how far it went, because what happens after 2 weeks is that the bell tissues start growing between the arms as they grow into medusa. So a small arm, remember, we removed 3 arms. A small arm growing in 10% of the animals 2 weeks after amputation is nowhere near what we are used to seeing in, say, planarian or hydra regeneration. However, this was enough to make us ask this question. Can we make ephyras regrow arms in the lab now that we know they have the capacity to do that? And by doing that, we hope to learn what are the factors that they send that activate their decisions to start regeneration. So this was a question that was tackled by this really talented graduate student then in the lab, Michael Abrams, and a very creative technician, Ty Bassinger. And then they agreed to do this. And perhaps they felt a little sorry about that because we spent more than 3 years of screening and screening and screening. Many factors failed. We screened all the usual suspects. I came from developmental biology background, so I screened a lot of signaling pathways, modulators of signaling pathways, and it didn't work. We tried some environmental stimuli, temperature, salinity, and density of the animals. We tried everything we could get our hand on, and it's just a lot of things failed, including some of my favorite signaling pathways, the WIN, EGF, and TGF-beta, pathways that have been shown to be involved in regeneration. But then, fortunately, few things did work. First, we identified a necessary condition, and that is activity. So we screened many geometries of the aquarium. We screened a plate, a beaker, and also cone shape. And the one that worked the best is what we call the cone butler setup. So this is a sand setting cone that was adopted for raising ephyras. And to each cone, we put aeration that serves to put oxygen into the water, but also what it does is that it generates current. So then instead of just settling in the bottom, what the ephyra does is in stagnant water, it simply settles to the bottom. In this setup, they continue to just flow along the current, or they coast along, or they swim intermittently. So in this condition, we found 3 things that can strongly promote regeneration in the lab. With any one of these 3 factors, first, we see that a fraction of the animals still regained symmetry. This was the most robust response to injury. But then we start seeing what we saw in the natural habitat, a small arm growing. And then 2 arms. And then better looking 2 arms. And in some cases, very few, you see very few of this, quite good looking arms that that's hard to distinguish with the original arms. If you have any quick questions, please feel free to ask along the way. We verify, yes, yes, okay. Yes, I see it, yep.
I'm wondering about the sort of rare regeneration phenomenon. Could that be a rare, atypical attempt to create symmetry? So you interpret it as a regeneration, but in fact, it could be an issue. You know, it's a failed process, and when you induce animals to do more physical activity, that's enhances the process.
Yes.
What I guess I'm saying, it's not regeneration per se. It's perceived as regeneration, but in fact, it is an attempt to build something asymmetrical that has gone wrong.
Yes, that is, there are many ways to interpret this. As you can imagine, we have exciting discussions with our reviewers on how to interpret what we see. We don't know what the answer is. One of my dream experiments, I would go to different localities where the moon jellies are found. So we have quite a few water places where we know the moon jellies reside, and then try doing those experiments in different places and see to what extent is there like a place in the natural environment that promote this response more versus others. And then maybe in some places when the environment is just right, it actually proceeds to full regenerations. But yeah, I agree with you in the sense that I think they just want to recover radial symmetry. That's why the most robust response that we see is no regrowing of lost arms at all, but just recovery of pure reorganization. Yes, and I see two more questions.
Yeah, I'm just curious whether the regeneration is due to the cutting, how you cut it. If you cut deeper into the tissues, and less regeneration. Maybe there are some cells that are more competent to regenerate or proliferate. In some cases, even move on.
Yes, that's right. That's right. So that in the sense is like what we see in children as well as in mouse digit, where you cut matters. Anything above the first phalange, and I'll get to that in a slide later, can regenerate. Mice can regenerate the digit tips, but nowhere. Not in response to amputations below. So we tried to do that kind of amputations too, trying different planes. Yeah. And then even like with the centimeters of millimeter precisions, my students were trying different things. And in fact, if you just cut like the very tip of their appendages, they can regrow that tip. So just like what we see, but then anything deeper, they stop regrowing. That's right. And then I think maybe I'll wait. Yeah. Yes. One more.
Oh, sure. Okay. I have two questions. When you did the experiment at Long Beach, you couldn't see the full regeneration because they were metamorphosing, I guess. And did they just not do that in the lab?
Same thing in the lab. So yeah, in the same time scale, about 2 weeks, they start just growing built tissues, and then they don't regrow what was regrown before. Yeah, so it's like we cannot get them to go further.
And then the other question I have is these images of the ephyra growing first one limb and then two limbs. Are those time courses? So it looks like the jellyfish first decides to grow one limb and then.
Oh no, they're different. That's right. Yeah. So you look at 20 animals, they regenerate to different extents. Some regenerate one, some regenerate two, very, very few that regenerate all three. And when I say
when they regenerate two, they do it at the same time. So
yes.
do one and then
no, that's right. We looked into that question. And when I say regeneration here, I'm being very generous, right? Because usually we associate regeneration with full recovery of functions or structures. So just keep that in mind. Yeah. Okay. All right. So of course, we see the arms, we verified that tissues are rebuilt in the right places. So this is a Palladian state that is enriched in the muscle. This entire arm is new, and you can see the growing ends. And then here's another movie. This is a recovering ephyra. This is about 2 weeks after amputation. So you can see these two are the new arms. And they recover synchronous pulsation, which means that muscle and neurons are connected functionally. All right, So what are the factors that promote regeneration? 3 things, and then there's a 4th one after this. Okay, so the first factor is as simple as it can get. A lot of food. That's it. But remember, this has to happen in a certain geometry. That's why we couldn't see this effect earlier in our earlier one. So what I'm plotting here is that it's each blue and gray pair is controlled and treated, right? So the control is just low food, and we set low food control to give us about 5 to 10% baseline regeneration to mimic what we saw in natural habitat. And then each one is an independent experiment from different batches of animals. Okay, so 3 times this is just a lot of food. The second one is insulin. So we use human recombinant insulin because that's what we can buy and mix that in the seawater, use artificial seawater, and that itself can recapitulate the effect of high nutrients. The third thing is reducing oxygen level in the water. Yeah, and this is about 50% reduction in the oxygen level. We flow in nitrogen. We also try different gas, and it has the same effect. So it really is reduction in the oxygen level that coaxes the animals to begin regenerating. So then we ask, well, high nutrients can be recapitulated by insulin and hypoxia, but we also want to see if there's a certain nutrient factor that can recapitulate the effect. We reason that the jellyfish are carnivorous. They eat a diet high in amino acids. So we tested branched chain amino acids that are known to be the anabolic class of amino acids. In particular, leucine, just leucine alone can recapitulate the effect of food. So 4 independent experiments, we verified that these are statistically significant. And also what I don't show you is that we have also characterized the number of arms, how long the arms are, and how well it recapitulates the morphology. And the treatment does improve. So increase in frequency of regeneration also correlates with improvement in the quality of regeneration.
Is
Josh?
it exclusive to leucine, or the other branched chain amino acids have some effect as well?
You're going to see some other amino acids. The reason we choose leucine because, okay, quick answer is we haven't tested other BCAA. The reason we went straight to leucine is because leucine is not only amino acid nutrient, but it also is a signal to some of the pathways in the growth pathways in the animals.
More can you put the location you put the amylase and then
Yes.
of that will reveal very strong.
Yeah, that's right. So we observe the animal after we yes. So at first it's open wound. You can see the gaping wound, and I usually show that, but I don't have it with me today. And then the wound, you can see the wound closes within 12 hours. Yeah. Yeah. Yes.
So I wonder how consistent this regeneration can be if you recut the jellyfish that you go with 4 arms and then you do it again.
You do it again. We have not done that experiment yet. We want to do that. And the reason is because we check regeneration for 1 to 2 weeks. That's when we can consistently see them. And then they start growing, maturing into medusa. But we can manipulate a little bit the timeline. I think we can do that. Yeah, we'll try that.
I have a question that kind of follows to the question that Robert asked me, because my reflection is in clinics when the kids in a very young stage, when they have to cut on their tip of the finger, looks like my reflection of regeneration is better when you actually keep peeling the thermal slave down to expose the whatever they call the
Yes.
to generate this structure. Like, do you see there's a correlation between the animals that are generated in your experiment regards to how the healing process looks like?
Yeah. Yeah, that is a very interesting point you brought up. And that is the reason why the regeneration, the fingertip regeneration is so controversial, because at least in the studies where it was reported, there are 2 requirements. First is that you cannot stitch the wound, right? Yes. And then 2 is you cannot use antibiotics. So that's why, and those are regular things that we do when kids get, well, yeah, finger injuries. So that's the reason. The quick answer to your question is no, we haven't looked at it specifically. But of course, obviously, we're not stitching, so we let the wound just expose to seawater. So they get placed back in the seawater and their aquarium and swimming naturally. So I think this mimics not being stitched, just being exposed to the wound. But that's a good question. We haven't looked at that more details. All right. So in summary, in the jellies, we found 4 factors that promote initiation of appendage regeneration. One is abundant nutrient, which can be recapitulated by leucine, insulin, and reduction in the oxygen level in the water. All right, but these 4 factors, yes.
How long will the jelly have the ability to regenerate a new arm? If they don't generate it in 1 week, and then regenerate the arm 1 month later?
We have not tested the age effect. Oh, but here's something that can get to that question is we've tested this in the medusa stage and then observed similar results. We haven't done it as systematically, but yeah, that's a good one. The age effect, definitely an important factor.
For me, you mentioned antibiotics. If you add antibiotics, does it inhibit regeneration of your polyps? Is there any microbial component to this? Is what I'm looking for.
Yes, there is. And that is, I was not planning to get into that in this talk, but our subsequent after the papers that you guys read is that we found the role of microbiome. And the reason why that is the follow-up project in the lab is because it seems like it's a strong effect. So because we're talking about nutrient and microbiome is at the forefront of nutrient metabolism, the boundary between us and food, maybe we think it might be one of the rate limiting factors. But yes, the quick answer is we put antibiotics, we see enhancement, certain antibiotics enhancement in the regenerative response. Okay, and Nafi, and then, yeah.
So was electrical stimulation or melatonin tried for arm regeneration?
Melatonin.
Or electrical stimulation?
I have not tried electrical stimulation just because that is more like what Michael Levine would do in terms, and I love all of his work. But no, I've talked to him about maybe trying some ion channel inhibitors, but we've not gotten to that just because there are so many things to try, not because it's not interesting. We have not tried melatonin, but my student is now a postdoc at Berkeley, and he is continuing the jellyfish sleep study and exploring if there is any relationship between sleep and regeneration. That's one of his side projects. Yeah. Okay. So high food, leucine, insulin, oxygen. What we notice is that these are just basic conserved factors in physiology. So that made us wonder if they could work as well in other poorly regenerating contexts. So to pick a poorly regenerating system, you would think that it's an easy task, but it's actually not. To really find a system, established system of poorly regenerating system is not easy because most of the regeneration literature reports positive results, which is presence of regeneration, right? And so the literature is somewhat silent on what not regenerate. So, but fortunately for us, we know that established models, some of the established model organisms in the lab are poorly regenerating. So this challenge is what is tackled by this team of fantastic graduate students, Fei Tan and Yutian Li, and really, really smart technician, Martin Hythe. And then at first they tried C. elegans. We're looking for small animals. First, they tried C. elegans, they tried halving C. elegans, but unfortunately, all of the halved worms died of hemorrhaging. So we encountered technical difficulty. We think C. elegans just don't have the right musculature to quickly constrict the wound, or they don't evolve to being halved, I guess. But then we got more lucky with another small model organism in the lab, which is the fruit fly Drosophila. So Drosophila belongs to this vast group of whole metabolous insects, which includes insects such as beetles, butterflies, and ants. And as a group, they don't regrow limbs as adults. Yep. Unlike if you're thinking of crickets and cockroaches, those are hemimetabolous insects, the other large group of insects, and they can regrow appendages upon molting. That's the key. Yep. But we know from studies from Hariharan's lab that as larvae, they can regrow imaginal discs. So that made us wonder, maybe they have the ability and maybe that can be promoted in the adult. Okay. So in Drosophila, we use specifically leucine and insulin. And the reason is because food is heterogeneous across animals. So we don't want to worry about what different foods are required, food factors are required. But leucine and insulin are specific factors. So if you think of your own leg, here's a schematics of a fly leg, and here's the femur, think of your upper thigh. The tibia, think of your lower thigh, and the tarsal segments, which corresponds to ankles and feet. And in the lab, in this experiments I'm showing you, we always cut across the tibial segment. Okay. Here's how we did the experiments. So the amputated flies get put back into the vials. And some of you who work with Drosophila, you know, they live in vials with food in the bottom. Okay. And then the control is just a regular lab food. So they go back, they come from lab food, back to regular lab food. And in the treated condition, we mix into the top layer of the food, leucine, insulin. And in this case, we added glutamine because it's known that in fly and mammalian cells, glutamine promotes leucine uptake. So my student decided to put that into the mixture. And we examine each fly every 1 to 2 weeks, all the way up to 3-4 weeks, however long they live. So we examine the tibia. So if the tibia is either cut, then we score them as no regeneration, or we saw elongation in the tibia. So in this case, it's just a schematic of a tibia that would be regrown fully. So here are some of the images of the control flies. Here's fly 1. Here's like 0 DPA. That means we cut them and then we took a picture right away after cutting. And then here's a picture of the same fly, that tibia, 3 weeks later. So you can see the control flies don't grow. Here's another fly right after amputation, 2 weeks later, and 3 weeks later. Okay. And here are some examples of the treated ones. Here's one fly right after amputation, and 2 weeks later. And here's another fly right after amputation, 2 weeks later, and 3 weeks later. Yeah. We quantified the result. So here it is plotted on the y-axis is change in the length of the tibia over days after amputation. So here's each line is the fly. So that's 116 flies, and this is in the control group. And here's in the treated group. Again, each line is a fly. And so as you can see, there's change in some of them in the length of the tibia over time. And this is an experiment that was done by a group of 5 students, including myself, because it was a lot of work, as you can imagine, to track flies and to take a picture of them. But it's basically what my point being that this is the results from independent experiments with multiple experimenters. We verify that the 2 distributions are statistically significantly different. And if you take the confidence interval of the control as your noise in the measurements, then you can determine that 50% of the treated flies show growth that is significantly different from the control. Hi. Yes.
I'm curious if you also measured the uncut limbs in the control and treated samples. So it looks like in the images that you showed on the previous slides, like the flies in the treated conditions were getting it quite a bit bigger, maybe because they're uptaking nutrients better or something like that.
Oh, you mean the body size? No.
Yeah, and I'm wondering if then, in addition to that, if their limbs are also just growing, even uncut limbs might be growing because they're eating better with the glutamine in the media or whatever.
Yes. So the fly is a size determinant. It's a size determinant. That means that after larval stage, when they hatch, the adult don't change size anymore. So that's why a lot of the growth study in flies are done in larvae. So you see bigger or smaller flies because of the differences in the larval behavior and feeding and processes. So we can't assess the effect of size on that. But
I mean,
well,
you have like an internal control because you cut 1 out of 6 limbs and then you have 5 other limbs that you can also measure. And if those are staying the same, then it's specifically regeneration rather than just limb.
growing.
Yeah.
That's right.
Swell.
Yes, that's right. So we've done that analysis. Exactly. We like that setup because even within the treated ones, some show change the limb length and some don't, and those don't. Yeah, those are basically this black lines here that you see. That's right. But to get back to your point, what we're studying to quantify if instead of body size, what we're measuring now is the lifespan. All right. So that's later. Hi, yeah. Alexandra.
Yeah, just a quick question. So you had some lines end at day 5. So what happens with those?
That fly died.
Oh, it just dies.
Yes. It's just random effect experiments that we can't control.
So that's interesting because there's a lot of them that end at like day maybe 12.
Yes.
Something like that.
Yes.
It's interesting they regenerate.
And some of them just cut short and experimental, but there's also ones that grow long.
Yeah.
That live longer. Maybe I'll take one more question. Maybe.
So
Yes.
it's fantastic that your essay is so noisy because that is a great opportunity to analyze the individual animals.
Exactly.
Find better for the proteomics. Just not only control versus treated, but what is the difference between the fly that regenerates and the fly that are treated but don't?
Yes, exactly. So one of the first things that we've done after this analysis that you see in it is to do RNA-seq. Yes, between, like you said, we've been treated one, so it's really clean, right? It's all the same condition, the ones that don't regenerate and the ones that regenerate. And the cool thing is like in the next slide, I'll see, you can tell, even though this measurement is 7 week scale, you can tell as early as within 12 hours which flies are likely to grow because of this next slide. Okay. So I've told you, when you look at this distribution, about 50% of the flies show growth, right? It's to various extents. So if you look at the pictures, it's nothing as dramatic as regrowing a whole limb, but it's beginning to do something. That's what we're keen on in the lab to study. So 50% show change in the length. In 3%, we show a joint reforming. So this is an uncut tibia as a comparison. Here's the control. So you do see that scab at the end. That is a normal wound healing. That is what we expect from wound healing literature in Drosophila. You have a wound, even if it's just epidermal wound, you develop scab. Yep, the scab is really clear, shiny and black. And then it's also correlated with inflammatory state in the wound area. All right, so here's the few ones that dramatically grow. Join, you can see here, the tip is very different. And this is actually one of the first ones that we found in the lab. As you can see, this samples, it's rare, but it's there. And to answer the early prediction, the ones that show regrowth also, as you see the end, they show modified wound healing response. They're scabless. And we know this is not just because the scabs peel off, because we track them from early on. So they never grew scab. So we think there is definitely modulation in the inflammatory response to wound. that is correlated with activation of more regenerative response. Okay. So yeah. Then we verify that yes, the tissues are there. So this is still ongoing, and this is some of our preliminary results. We're using muscle fluorescent reporters. So in this case, we have two going right now in the lab. One is MHC-GFP, which is a myosin heavy chain promoter, which drives, which is enriched in the skeletal muscle, and that we're using the promoter of MHC to drive GFP. Another one is we're using the promoter of MEF2, which is a classic muscle marker and driving a fluorescent protein. So here's a representative image of what the residual tibia looks like after amputation, the fluorescent signals expressed in the muscle. Here's the control limb 3 days after amputation. Here's a week after amputation. Here's now a treated one 3 days after amputation and a week after amputation. So we are obviously doing longer term imaging now, and we're trying to clean how much we can see just from the muscle signal alone, which part is the new growth, which part is the existing one. But so far, what we're seeing even in the short time scale is that the muscle is preserved better in the treated residual limbs. All right, so here's just a close-up of the 7 days post-amputations in the residual limb, just comparing that to the uninjured. So you can see it loses tension, because we cut right in the middle, but it recapitulates it retains the spatial asymmetry. All right, so then emboldened by results in jellyfish and in flies, we ask if the same strategy might work in mammals. And there, as I touched upon earlier, is we go to study limb regeneration in mammals. What we will study is the mouse digit system, because it's just a gentler amputation. So in the mouse digit, this is just a beautiful system that was developed by Ken Munioka in Tulane. So we owe a lot of what we can do to a lot of basic works that he's done over the years. So we see that in the mouse digit, the tip regenerates spontaneously. But any cut below the first phalange, and the convenient morphological marker for that is just the nail. Any cut below the nail does not normally regenerate. I'm going to show you bone staining as well, where you can see more precisely where the boundary of regenerating and not regenerating lies. So then the big question driving this studies in mouse digit is, how can we stimulate regeneration from the proximal cuts as a way of understanding, can we potentially someday ever think about inducing limb regeneration in humans? Right, so then we try. So in the mice, we perform the proximal cuts in the regions that normally does not regenerate. And then we administer leucine and insulin. So here's how we do the experiments. In the control mice, we simply give them water, drinking water as usual. But then in the treated mice, we give drinking water supplemented with leucine and glutamine, just like in flies. And then instead of giving them insulin, because insulin is proteolytically digested in mammals, that's why we cannot take insulin orally. Reason that, well, let's give them just sugar, because sugar would naturally induce the production of endogenous insulin. And we researched the literature and we determined the level of sugar that would induce increase in insulin just at pre-diabetic level. So it's about 3 times the amount of what they normally get, the carb that they normally get from their diet. So this is an important step. The digit part removed was fixed right away, so we know how much we're removing. And then we image the digits after every 1 or 2 weeks or so. And then we dissected the digits at week 7 and 8 for to stain for the bones. So here's some examples. Here's some of the digits, 7 weeks post amputation. Here's the control paw. So you can see we amputate across digit 2 and 4. We can see where the digits are shorter. And here is some of the more dramatic ones in the treated, on the treated mouse. So here is a digit 2 that is regrowing the distal phalange. And we even see growth in the other digit as well, even though not as dramatically as the second digit. And we see response to various extents, not all this dramatic, but we see activation of regenerative response in about 20% of the digits we analyzed. A more precise way to assess regenerative response is by looking at the bones. So if you think of finger bones, we have 3 digit bones, right? The distal one, just call that P3 for short, and the middle one, P2, and that's what we're concerned about here. Here is an example of a bone staining from a control digit that was cut across the distal end of the P2 bone. Here's the part removed. You can see the entire P3 bone, and then the tip, the end of P2 bone. And the one labeled S is an important one. It's a sesamoid bone. And in our anatomy, it's part of our wrist. It's the small bones that are that provide mechanical integrity to our wrist. Here's the digit stump, 7 weeks post amputation. So you can literally put the part removed to the stump and then we recover the digit. Okay. Here's 2 of the more dramatic ones from the treated. In this digit, this was amputated across the proximal part of the P3 bone. Here's the part removed. And here's the digit stump 7 weeks later. So as you can see, it has regrown the P3 bone. And if some of you notice, it has more of this weaved look. And this is actually what's also observed in the spontaneous digit tip regeneration. The bone is different. And the thinking there looks different, but mechanically, they're doing more careful testing. Mechanically, they think it's functioning the same way. Here's another one that's more dramatic. This one was cut across the distal end of the P2 bone. So here's the part removed. The entire P3 bone is removed. The epiphyseal cap of P2 and also most of the sesamoid bone. And here's the digit stump 7 weeks later. So we see that the P2 bone has been regrown and the beginning of what we think is distal digit bone and also the sesamoid bone has been recovered as well. So in summary, we find that appendage or limb regeneration across animals can be coaxed to start, at least in some animals, using nutrient factors. Yeah, and this was, if you want to see more details about the study, we published this in 2021, at the end of 2021. So first, of course, in the lab, as you can imagine, we have more questions. We have first is that regeneration is not complete and it doesn't occur in all animals that we treat. So why is that? So we keep screening for more synergistic factors, yeah, that could enhance the effect of leucine and insulin. But also how do seemingly simple nutrient factors can promote regenerative response? So we're just trying our hands in all methods that we could try. But then for now, though, what we think is happening, so right around the time we published this work, we found this paper from Andrew Will's lab. So rock tadpoles can naturally regenerate their limbs, except for a couple of days period after, right around in the beginning of of their life stage, where it's called a refractory period, where they used up the yolk from the mom and start making their own, start feeding on their own. So for this couple of days, refractory period, they stop regenerating limb. So Andrea Wilslap found that just by extra feeding, they can rescue the refractory period. And then when we broaden our search to beyond limb and appendages, we found that ctenophore, this is what was from Mark Martindale's lab in 2017. So ctenophore comb jellies regenerate very well. Being half, they can regrow the missing half. But then they found that when they put them in poor nutrient environment, 50% of the animals stop regenerating. Exactly. And instead circularized, just like what we see. So we think that what is happening across systems could be, in some systems at least, that food or nutrient environment is a cue to regeneration decisions. So the hypothesis that we're pursuing in the lab now is that could it be that energy allocation influences the decisions to regenerate? All right, so with that, again, the students who did the work, Michael Abrams, now postdoc at Berkeley, Fei Tan and Yu-Tian Lee, who did the work in flies and mice. And then we highlight our collaborators, David Gould, now at UC Davis, and John DePere at Caltech. And then the many seed fundings we get from Caltech that funded this project, as well as philanthropic gifts. Thank you.